Um, I'm a student who is doing biomedical laboratory science in Rwanda, the Ines Ruhengeri. I'm going to present uh, some more topics in applied immunology. Seeing Ines Ruhengeri, Faculty of Health Science, Department, Module 3, names no number. We're going to talk about these topics: immune checkpoints, cytokines, monoclonal antibodies, checkpoints inhibitor therapy, CRT cell therapy or chimeric antigen receptor T cell therapy, immunotherapy. So let's start with immune checkpoints. They are proteins that regulate how the immune response is. Immune checkpoints are regulatory pathway in the immune system that maintain self-tolerance and modulate duration and intensity. Maintain self-tolerance and modulate duration and intensity of immune response to minimize damage to health tissue. They are primarily inhibitory signal provided by molecules such as programmed death ligands and even cytotoxic T lymphocyte associated protein 4. Immune checkpoints are these. They are to maintain self-tolerance and modulate duration and intensity. These are seen here. There is figure that help us to somehow to show. Here you are seeing antigen presenting cells in tumor tissues. It is here as you see. Make this yeah, this one. These are antigen presenting cells which is found in the tissue. If these antigen presenting cells detect the antigen, it processes it and present antigen to the T cells. This cell has receptor of CTLA4, it has CD28, and even it has T cell receptor. This is antigen presenting cells which present antigen to T cells. This should be MHC, major histocompatibility molecule, help in a presentation of antigen to T cells. And this is T cell receptor. When it join, this is the first signals. Then after, second signal is between between C. It is between B7 protein or CD86 or CD18. Here you see the step by step. This was the first step. Second one, this. Then this is antigen presenting cell that are processing the antigen. You see. Third step, lymph nodes. They migrate. It is from tissue to the where the T cell is found. When here it is presenting, here there is the MHC presenting to T cell receptor, these first signals. Then second signal should be between CD28 and CD80 and 86, which together are called B7 protein. These are helping when they make it second signals, they allow the activation of B cell and T cell. First T cell is directly activated. After activated, it migrates to tumor tissues and even it releases cytokines. When it is activated, there is immune cells. Here there is this programmed cell death ligand 1 and programmed cell death 1. These signals help to attack the cells, the tumor cells, in order to destroy it. Yeah, this somehow how the immune checkpoint. Remember above we have seen immune checkpoint examples. They are here. This one. First one is PD1, PD. These both of three help to regulate how the immune response should be gone. And then why are they important? They prevent autoimmune disease by ensuring immune system does not overreact to normal tissues. Then cancer cells can exploit these checkpoints to avoid being destroyed by immune system, which is why they are targeted in cancer immunotherapy. Then second topics about cytokines are small signaling proteins. They are protein also, even I have seen checkpoints or are also protein. These are small signaling proteins secreted by immune and other cells that regulate immunity, inflammation, and hematopoiesis. They act as messenger to modulate behavior of immune cells. Here there is a schedule that can explain it. When the antigen presenting cells present antigen or another antigenic substance to T cells, then cytokines released. These are tumor growth factor, beta, is interleukin 6, is interleukin 4. These are released in order to trigger another cells. There is this T helper 1, this T helper 17, this T helper 2, this T regulatory cells. T helper 2 help to activate B cells. with, with the help of interleukin 4, interleukin 5, and even interleukin 13. These help to secrete immunoglobulin A for inflammation, C macrophage, and and uh, these cytokines are including interleukins, chemokines, and the tumor necrosis factor. Um, let's continue. Some function of cytokines are here. Pro-inflammatory cytokines, so there are cytokines that enhance response to fight infection or tumor. Anti-inflammatory, those are that stop the action of pro-inflammatory cytokine. Chemokines are another guide the migration of immune cell to site of infection. These chemokines, they mark the foreign substance, and then while immune cells are migrating, it responds to chemokines. Growth factor promotes cell differentiation and proliferations, like interleukin 7 stimulate T cell development. Cytokines ensure a coordinated response, balancing activation and suppression to maintain immune homeostasis. Then monoclonal antibodies. These are the laboratory produced molecule designed to bind specific antigen. In immunotherapy, they work by targeting cancer cells, immune modulations. There is a mechanism. These monoclonal antibodies are produced from they are produced from um, they are produced from hybridoma cells that are manufactured by combining normal cell and tumor cells. So during the usage of monoclonal in immunotherapy, uh, as I mentioned, it 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 has specificity to bind to its specific antigen. Here, monoclonal antibody can bind to specific marker on cancer cells leading to this destruction. According to this, the they do monoclonal antibodies called nivolumab, primporamzumab, and semiprimab, and distanimab. This map is main meaning monoclonal antibodies. Then they they are sent into the body so that it can mark the cancer cells. After that, while it has marked the cancer cells, it show to the body there is foreign substance and help in activation of T cells, which are responsible to release cytokines and even to destroy those tumor cells. As you are seeing here, T cell activation, there should be co-inhibitory signals, and even we have when this antibody introduced should block this channel where the tumor cell pretends to be a normal one by using the checkpoint which you have seen before. Then after, can continue by principal checkpoint inhibitor therapy. Checkpoint inhibitor therapy target immune checkpoint molecule to unblock the immune system, allowing it to recognize and attack cancer cells. Cancer cells exploit immune checkpoint to evade immune surveillance. Here, as I have mentioned, this is where the immune checkpoints are occupied by tumor cells, and then tumor cells pretend to be a normal one. Blocking these checkpoints restore the immune system's ability to detect and destroy cancer cells. Checkpoint inhibitor like nivolumab and ipilimumab have revolutionized cancer treatment by enhancing T cell activity against tumor. This is how treatment by using checkpoints inhibitor is applicable. As I have mentioned, in a checkpoint we have PDL1, PD1, and even a CTLA4, or cytotoxic lymphocyte associated protein 4. When the cancer cells copy the character of checkpoints, we have to make the things that can stop the action of checkpoints, and that product we have manufactured is called checkpoints inhibitor. This nivolumab and ipilimumab are those called checkpoints inhibitors. They try to block those chain where the tumor cell have pretend to be a normal one, and those inhibitors stop that chain. When the tumor cell have created like these things, then novobiocin come to break this and stop the pretending of tumor cells. After it block this chain, then the body realize that there is foreign substance, foreign cells, and then T cell can be ready to attack those tumor cells. There is here also we can use chimeric antigen receptor T cell therapy. Here we have another immune therapy application, which I've seen chimeric antigen receptor T cells. What are the chimeric antigen receptors? Therapy involved engineering a patient T cells to recognize and kill cancer cells. During the manufacture, these are also antigen or genes manufactured in a laboratory where they take blood from patients in order to get T cell. After they get blood, They separate in order to get T cells only. Those T cells are carried into the lab and for making chimeric antigen receptor T cells in the lab, where they take those T cells and add a specific genes to that cell. That gene are able to take and are able to bring bring characteristics to the T cells in order to bind the target cells or tumor cells. Here they insert genes from RAP and then T cells have been modified into those genes are called chimeric antigen receptor because they add a gene which is receptor in order to attach to that tumor cells. Then after they inserted the gene, they culture in culture media, they culture T cells. It may took several weeks in order to get sufficient T cells in order to treat it. After they culture, before they introduce those cultured T cells, they should make chemo- chemotherapy. Those chemotherapy are 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 having purpose of decreasing the capacity of existing immune system to react to that new T cells. After they weaken the T cells, uh, cells or capacity of cancer to attack that T cells, and they introduced T cell and found there is a good environment to adopt. And after T cell is adopting, then it can become killer tumor cells and then distract the tumor cells because it has capacity to bind to that and it has got time to change or to adapt. So these are how chimeric antigen receptor T cells therapy are worked. Then risk of CAR T cells therapy. There are cytokine release syndrome. These are disease, neurotoxicity, on target of tumor effect, infection, graft versus host disease, and how immune therapy used to treat autoimmune disease. These are some approach. Immunotherapy of autoimmune disease aim to modulate the immune cell to reduce self-reactivity without compromising of activity. Approaching include monoclonal antibodies can use to neutralize cytokines or immune cell-driven inflammation, checkpoint inhibition, or checkpoint modulation, controlled tolerance therapy, B cell depression. Cancer cells can be treated by using chimeric antigen receptor, target cells, surface antitoxicity. Here we can use also immunotoxic immunotherapy or immunotoxins are also therapy used where they take monoclonal antibody and they add toxin like Shigella, diphtheria to that monoclonal. That then monoclonal are introduced into the body to attach to the tumor target cells. After it attach, then the toxin are released to attack that cell and destroy it. Here, autoimmune disease can be treated according to this immunotherapy. There is immune checkpoint inhibitor and even evaluation management, patients with cancer, and cortisol and other immunomodulatory treat agents. Those are things that can be used to treat the patient with immunotherapy. So thank you. I hope this content have helped you. So thank you for following, and you can give this video to your colleagues so they can study well. Thank you.